Hello everyone, and welcome to the first episode of Pigeon Genetics. In this first video, I'll be explaining the purpose of the series, and then I'll give a talk explaining many of the terms and background information that will really help you understand how genetics work in pigeons, and how to use it to your advantage. I'll put up a screen now with the timestamps where we talk about different terms, just in case you're returning to the video to clarify or go over something again. Also, please make sure you're subscribed so that you don't miss a future episode. Okay, I'd like to make it very clear from the beginning that this series is aimed at hobbyist pigeon fanciers and backyard breeders. This is not going to be a purely scientific study of genetics. I'm not going to be telling you things like recessive red is caused by a mutation of the SOX10 protein gene. If this is the information you're after, you're best to dive into the scientific literature written by people much more qualified than I am. Instead, this series is designed to give you the framework to understand genetics, and to teach you how to breed the specific colours and types of pigeons that you're after. I must warn you that this first episode will be quite long. I'm going to be giving you a lot of background information. Please don't get discouraged if it doesn't all make sense immediately. Feel free to pause, to rewind, and to listen as many times as you need. And if something still doesn't make sense, please feel free to ask a question in the comments. We are going to be going over a lot today, and there are some points that may require further explanation. For some of these points, I'll be making supplementary videos to explain them more fully in the future. I know most of you are watching this series because you'd like to know how to breed specific colours, like barless or almond pigeons. And this information will certainly be coming soon, but there will be a few videos of background information first. I'd really like to encourage you to make sure that you have watched this video and those that are coming up, because having an understanding of these underlying theories and principles will really help when it comes to practical breeding. Remember, this can be a complicated topic, and you're not expected to understand everything instantly. The more time you spend listening and learning, the easier it will become. And always, feel free to ask questions. Now, let's get into it. For a beginner, the jargon and terms around pigeon genetics can seem quite intimidating. But they don't have to be. Remember, pigeon genetics is basically just a set of instructions on how to build a pigeon. So we can use a simple metaphor to try and understand it. Imagine that for every pigeon, you have 2 books, with each book containing every instruction you'll need on how to build your bird. One book of instructions comes from each parent, and together, they determine the makeup of the new baby pigeon. This set of 2 books is called the genome. The genome is all the genetic information that makes up the organism. Now remember that I said a 2-book set. I'll explain that further shortly. Inside both of the books, you have chapters. In our metaphor, these chapters correspond to the chromosomes. Pigeons have 40 pairs of chromosomes, obviously making a total of 80 individual chromosomes. Imagine our books again. Each chromosome or chapter in one book has a matching partner in the other book, for a total of 40 chapters in each book. Remember, the chromosomes in each pair come from opposite parents, one from each. The chromosomes are where the instructions for building the pigeon are recorded. Things like eye colour, wing pattern, and beak size. Each matching pair of chromosomes carry the instructions for the same thing. However, they do not always have to agree. For example, the instruction for eye colour is in the same chapter in each book, that is, on the same chromosome. But one chromosome might say, produce a pearl eye, while the other says, produce a yellow eye. We will discuss what happens in this instance later on, but for now just remember that our instructions are organised into specific chapters called chromosomes, and each chromosome has a corresponding, but not always completely identical pair. When the bird is developing in the egg, these instruction pairs are compared against each other to determine what the baby pigeon will look like. Inside each of our chapters, you have the individual sentences. These are the specific instructions for certain traits. In our genetic context, this is called the locus, or as a plural, loci. Remember I said earlier that the instructions on each chromosome don't always agree with each other. The instructions in each book might be different. At each locus, there are alternative instructions that could be there, and these are called alleles. To give an example in the context of a pigeon, one of its chromosomes may be coding for a check pattern, while the corresponding pair is coding for a bar pattern. They are both alternative patterns, and therefore are alleles of each other. Compare this to something like bar pattern and head crest. These are totally different and unrelated things, not alternatives, and therefore not alleles of each other. Generally, from the point of view of a hobbyist, when we say gene, we're usually talking about either a certain locus or an allele at a certain locus. The pattern gene is the position on the chromosome where the pigeon's pattern is determined, whereas the checker gene is the specific code for the check pattern, which is different from the code for bar or barless. This is more broad than the strictly scientific definition, but remember this series is aimed at hobbyist breeders, rather than scientific professionals. So in our context, this definition is generally acceptable. Before we move on, we need to spend just a few more moments talking about the chromosomes. We already know that there are 80 chromosomes in total, made up of 40 matching pairs, and each of these pairs contain instructions for certain traits. Now that we understand what a chromosome is, it is important to know that there are two different types of chromosomes. Of the 40 pairs, 39 pairs are known as autosomes. These are the standard, normal chromosomes. The one remaining pair are the sex chromosomes, or allosomes. In the hobbyist context, you'll likely never hear the word allosome. The terms sex chromosome or sex-linked are much more common. You will often hear genes on the autosomal chromosomes described as autosomal genes, or simple genes. This just means that they work independently of the sex of the pigeon, and each bird has two copies of the gene, one from each parent. The sex chromosomes are different. Instead of matching pairs like the autosomes, there are two different sex chromosomes that pair together. In pigeons, these are called the Z chromosome and the W chromosome. It is this pair that determines the pigeon's sex. Hens have a ZW pair, whereas cocks have two Z chromosomes. Because of this, we are able to have what are known as sex-linked genes. All our sex-linked genes are found on the Z chromosome. There aren't any known genes stored on the W chromosome. This means that there are some genes that cocks are able to have two copies of, whereas hens can only have one. Imagining our example of two books again, this means that when it comes to sex-linked genes, hens will only have one set of instructions, without another book to compare it against. We will expand more on this later as our knowledge and understanding grows. Let's take a short break here to recap before moving on. We've already taken in a lot, but this is very valuable background information that will help us understand more moving forward. So far, we have learned that genes are stored on chromosomes, and typically each chromosome has a matching pair. The exception is the sex chromosomes, which determine the sex of the pigeon. We also know that there are sometimes alternative genes for various functions, and these alternatives are called alleles. Now, we need to learn two words that help us understand why a pigeon looks the way it does, and what we need to do to produce more like it. These words are phenotype and genotype. Again, these words may sound scary, but they're quite simple. Phenotype is simply what the bird looks like, whereas genotype is the combination of genes that make up the pigeon. The genotype is the book of instructions, the phenotype is the result of following those instructions. This is fairly straightforward, but there is a slight difficulty that sometimes arises. There are some phenotypes that can be caused by multiple genes. For example, there are actually quite a lot of ways to produce a white pigeon. It can go the other way as well. Some genes are highly variable in their expression, and can produce pigeons that look quite different from each other, such as the opal and almond gene families. This can prove to be a problem, but can generally be solved with accurate record keeping and breeding tests. We'll discuss these in a later episode. We have learnt a lot of technical things, so now we can move on to something you might find more practical. When working with genetics and breeding the many different kinds of domestic pigeons that are available, we are working with mutations. A mutation is a change, a variation, a difference. But a difference from what exactly? Well, a mutation is a variation from the wild type. So what does that mean? The easiest way to describe wild type is the typical, unmutated form of an organism. It is what a wild animal would look like if it was never selectively bred by humans. So let's consider what that means in the context of pigeons. The standard wild type pigeon is a blue bar pigeon, about the size and shape of your typical racing homer or feral bird, without any other colour modifications, no crest or feathered legs, no fanned tail. It does not tumble or roll. It is a standard, plain, unremarkable blue barred pigeon. It is this wild type that any mutation or variation is compared against. To be clear, it isn't always the entire bird that is a wild type. Each locus, remember, a locus is an individual instruction on a chromosome, has its own wild type, and may have mutant alleles that are different from the wild type. In genetic notation, the wild type is signified by a plus sign, but this will be more fully explained in a future episode. Okay, now we know the difference between a wild type and a mutant allele. Please cast your mind back to the beginning, where we talked about the 2 books of instructions. We already know that each of the 2 chromosomes come from a different parent, and that they do not always have to agree with each other when it comes to their individual instructions. We know that, for example, one chromosome can say, make a bar pattern, when the other says, make a check pattern. Now we're going to learn the terms we use to describe these scenarios. When the instructions for a specific gene on both chromosomes match, this is known as homozygous. When a gene is in the homozygous state, it means that both chromosomes agree on which instruction to follow, simply because they only have one instruction. They both say the same thing. The opposite of homozygous is heterozygous. This means that the 2 chromosomes carry different instructions for the one gene. When this happens, there needs to be a method to determine which instruction to follow, and we will go over this shortly. A simple way to remember this is to know that the prefixes homo means same, and hetero means different. Homozygous, same instruction. Heterozygous, different instructions. Before we carry on, you might have picked up that there is one issue here. What about the sex chromosomes? We know that a hen has a Z and a W chromosome. These are totally different chromosomes, so they can never match. What happens then? When a hen carries one copy of a gene on her Z chromosome, it is called Hemizygous. This is neither homozygous nor heterozygous. She simply has one Z chromosome, so there is nothing to compare it against. As cocks always have 2 Z chromosomes, we simply use the standard homozygous and heterozygous system for them, even when referring to genes on the sex chromosome. Now, when we have a situation where the 2 chromosomes, that is, 2 sets of instructions, differ from each other, how is it determined which instruction to obey? Well, that comes down to the dominance of each allele. You may have heard words like dominant and recessive before. We can talk about them now, after these quick notes. While an allele is usually measured as dominant or recessive against the wild type, alleles at the same locus are also recessive or dominant in relation to each other. Using the pattern locus as an example, Barless is recessive to Czech, but neither Barless nor Czech are the wild type. I'll explain this more fully in a future episode, but just try to remember this for now. Another very important point to remember is that we only compare alleles at the same locus with each other. If you need a refresher on what this means, feel free to rewind to that section, because it is very important. I've heard people say things like black is dominant over white, but this is not a true statement. These genes are not alleles. In fact, they're on different chromosomes altogether, and cannot be compared as dominant or recessive in relation to each other. An allele is considered dominant when a single copy of it is powerful enough to mask or hide whatever allele is on the matching chromosome. Let's think about Barless and Czech patterns again. A single copy of the Czech allele is enough to completely hide the Barless allele. The Barless gene is still there on one chromosome, and can still be passed on to offspring, but in this particular bird, it is totally hidden by the presence of a more dominant allele, that is, the Czech pattern. An important point to note is that a dominant allele cannot be hidden by a weaker allele at the same locus. A friend once asked me if his blue bar pigeon could be carrying the gene to make a black pigeon. The answer is no. The allele behind a solid black pigeon is dominant, so it cannot be hidden in a blue bar. This standard form of dominance is sometimes called complete dominance. There are some alleles, though, that don't neatly fit into this category. Some, like classic grizzle or indigo, will produce a different phenotype, depending on whether or not the pigeon carries one or two copies of the allele, that is, heterozygous or homozygous. Some people consider this to be a kind of weaker form of dominance, and use words like partial or incomplete dominance or co-dominance to describe these traits. There is a bit of conjecture around which terms are correct and when to use them, but for the hobbyist this isn't too important. Just remember that there are some genes that do express in the heterozygous state, but express differently or more fully when homozygous. The opposite of dominant is recessive. A recessive allele requires two copies, one on each chromosome, to express into a phenotype. A recessive allele will be hidden by just one copy of a more dominant allele. Using the pattern series as an example again, to produce a barless pigeon, you must use two parents who both carry the barless allele, and they both must pass the allele onto their offspring. If a young bird does not receive a copy of barless from both parents, it will not express a barless phenotype. Something that will get its own video, so I will not go over it fully here, is genetic notation. This is a useful tool we use when writing down our genetic records, or calculating what we expect in the future. In this system, each allele receives a letter as a symbol, with dominant alleles being noted with a capital letter, and recessive genes in lowercase. Now, I know this has been a long video, and we have gone over a lot of new things. There are just a few more for this video, then you'll have most of the groundwork and background information to start having a real understanding of pigeon genetics. Remember, you can pause, rewind, and go over anything you don't quite understand, and feel free to ask questions in the comments. Okay, we're almost done. Let's touch on linkage. Linkage will receive its own video for a greater explanation, but as a quick explanation in this video, you should know there are two types of linkage: genetic linkage and sex linkage. Sex linkage simply means the gene is stored on the Z chromosome, and is therefore passed along with the genetic material that determines the pigeon's sex. As of yet, all genes passed on the sex chromosome are found on Z. There are no known genes on the much smaller W chromosome, so generally we can just ignore it. Genetic linkage is similar to sex linkage insofar as it means two genes are stored on the same chromosome. The difference is this time it is an autosomal chromosome, not a sex chromosome. An example is the pattern locus and the spread locus, two genes found on the same chromosome. If you found the book illustration helpful, you can imagine that linked genes are separate and unrelated instructions, but they are in the same chapter of our books. Crossover is a very interesting phenomenon related to linkage. As you know, pigeons have 2 copies of all the autosomal chromosomes, and male pigeons have 2 Z chromosomes. You also know that these 2 copies can have different alleles on them. Crossover occurs during the creation of a new sperm or egg cell. As each parent is passing a copy of its genes to the next generation, the chromosomes they're passing on break apart and then recombine. So the chromosome the baby receives isn't an exact copy of the one that its parents held, but a combination of the parent's 2 different chromosomes. As an example, consider spread and recessive opal. These 2 genes are carried on the same autosomal chromosome. A pigeon may carry spread on one of its copies and opal on the other. If a crossover occurs, the young pigeon in the next generation will end up carrying both spread and opal on just one of its chromosome copies. Another term you may not have heard before, but may come across in genetic literature, is epistasis. A gene is described as epistatic when it covers over or hides other unrelated genes. An example is recessive white. When a pigeon carries 2 copies of the recessive white gene, it will simply be a pure white pigeon, regardless of what other genes it carries, with a few rare exceptions. The pigeon could carry genes for spread, or barless, or ash red, but this would all be invisible because it is being hidden by the epistasis of the white gene. This again exemplifies the difference between a pigeon's phenotype and its genotype. Without scientific gene sequencing, the only way to determine what genes are hidden under the coat of white would be to do breeding tests. Finally, you should know that some genes are considered lethal, either in the heterozygous or homozygous states. An example is dominant opal. This lethality means that young pigeons carrying the lethal genes typically do not hatch from their eggs, or die soon after hatching. Well done! I know we've gone over a lot today, and not everything will have sunk in, but now you have a basic understanding of the terms and phrases used in hobbyist genetics. Remember, you can go over anything you didn't quite understand again, and ask questions in the comments. I'll also be putting out more detailed videos, a few of the terms mentioned in this video, to make sure that everything is clear. There is a great community of amateur pigeon geneticists online, always willing to help new breeders. And if pigeon genetics is something you're interested in, I'd encourage you to get to know these breeders. Their expertise has been invaluable to me, and I'm sure you'll find the same. I hope you've enjoyed this video and want to stick around as we learn more about genetics together.